This video will talk about the processes involved in the electron transport chain. First, a quick overview of the electron transport chain. The processes of ETC occur in the cristae found in the mitochondria of the cell. Both the cristae and mitochondria have cellular membranes which consist of a phospholipid bilayer. The space between the cristae and the mitochondrial membrane is called the intermembrane space, while the space inside the cristae is called the matrix. Across the membrane of the cristae are multiple complexes. These structures are enzymes, or proteins, that perform a specific function in ETC. Now that you have a general overview of the components of ETC, it's time to get into the sequence of processes that make up the electron transport chain. NADH is an electron carrier that can carry 2 electrons, and is the first step to starting ETC. NADH carries its electrons to the first complex, called NADH dehydrogenase. As a result, NADH oxidizes into NAD+, hydrogen, and electrons, which are set free in the process. These electrons are transferred through complex 1, and in the process power the enzyme to pump hydrogen ions, or protons, from the matrix into the intermembrane space. This requires the energy that is given by the electrons since it goes against the current concentration gradient. The electrons that were in complex 1 now are transferred to coenzyme Q10. Next, FADH2 moves to complex 2. Because FADH2 bypasses the first complex, it produces less ATP than NADH since it uses one less complex. Again, it oxidizes into hydrogen and electrons, as well as FAD+. However, complex 2 does not have the ability to pump protons into the intermembrane space. The electrons set free from the oxidation of FADH2 are then passed from complex 2 to Q10. By receiving electrons from both complex 1 and complex 2, Q10 is reduced. It then gives its electrons to complex 3, which uses the electrons to pump more hydrogen ions across the phospholipid bilayer. This further increases the number of hydrogen ions in the intermembrane space, simultaneously increasing the concentration gradient. Then, complex 3 passes its electrons to cytochrome C. Finally, electrons continue from cytochrome C to the final electron acceptor, which is oxygen, or O2. As a result, more hydrogen ions are pumped from the matrix into the intermembrane space from complex 4. Additionally, water, or H2O, is created as a byproduct. This process repeats itself until a powerful electrochemical proton gradient has been created, where a large concentration of protons is present in the intermembrane space. Because of this, the hydrogen ions want to travel into the low-concentrated matrix, but can't. As a positively charged ion, they cannot cross the phospholipid bilayer. As a result, the only way they are able to move is through the ATP synthase. As the protons rush through the ATP synthase, they power the phosphorylation of ADP and inorganic phosphate. This process is called chemiosmosis. It creates approximately 34 ATP molecules, signifying about 90% of all ATP produced during cellular respiration. The entire process of ETC is oxidative phosphorylation. Oxygen pulls the electrons through the electron transport chain, pumping protons into the intermembrane space, which phosphorylates ADP and inorganic phosphate to create ATP energy. This is how the body is powered through cellular respiration.